Hello, and welcome to this webinar titled Advances in Mass Spectrometry for the Analysis and Bioanalysis of Antibody Drug Conjugates, hosted by Biopharma Asia magazine and presented by Dr. Arnaud Delobel. My name is Shri Patel, and I will be the moderator. Now, before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we'll go through these at the end of the presentation. Now, please allow me to introduce you to our presenter. After earning an engineering degree in organic chemistry, Arnaud Delobel specialized in mass spectrometry of proteins and peptides during a PhD at the CNRS in France and a postdoctoral at the University of Liège in Belgium. He joined Quality Assurance Assistance, SA, in 2006 as R&D analyst and occupied several positions until he became R&D director in 2016. I would now like to invite our presenter, Dr. Arnaud Delobel, to join our webinar. Welcome, Dr. Delobel.
Thank you, Shri, for this kind introduction, and welcome everybody. Before going into the heart of the topic, I will now say a few words about Quality Assistance. So Quality Assistance is a contract research organization based in the south of Belgium. For more than 30 years, Quality Assistance has been providing analytical services to the pharma and biopharma industry. More than 150 highly qualified employees work for over 100 worldwide companies in compliance with EMA and FDA regulation. Our main force is to have all the laboratories centralized on one site in Belgium, which facilitates project management. As regards our activities on ADCs, we combine an experience of more than 30 years on cytotoxics with 16 years experience in biologics. Last year, for example, we worked on 9 different projects with more than 5,000 hours of analytical work. For more information, please visit our website or contact me by email. Let's move now to ADCs. The characterization of antibody drug conjugates represents an analytical challenge due to the complexity of the molecule. In addition to the complexity of the monoclonal antibody itself, an additional level of heterogeneity is added with the conjugation of the linkers and drugs. All the tests commonly performed on monoclonal antibodies have to be carried out on ADCs, but other critical quality attributes have to be evaluated, such as the drug-to-antibody ratio, or DAR, which corresponds to the number of drugs conjugated to an antibody, the localization of the conjugation sites, and the occupancy of these sites. To solve these challenges, I will try to convince you today that mass spectrometry can be a valuable tool. So, how can MS be used for ADC characterization? It can actually be used at different levels. First, for the study of higher order structures, aggregates, or for binding. It can also be used for the study of the intact ADC. That's what is called the top level. The middle level corresponds to the study of subunits or fragments of the ADC, usually in the molecular mass range from 25 to 100 kilodaltons. The bottom level analysis at the peptide level can also be used. That's the peptide mapping approach. And finally, small molecule residues coming from process or free drugs can be quantified by mass spectrometry. The talk today will focus on top, middle, and bottom levels. If you want to have a good overview of the application of MS to the study of antibody drug conjugates, I encourage you to have a look to the review published by the groups of Alain Beck and Sarah Cianferrani last year in Expert Reviews in Proteomics. So, the webinar today will deal with 5 specific points. First, top-level analysis for DAR determination, analysis of ADC subunits, analysis of ADCs by peptide mapping for primary sequence confirmation, determination of conjugation sites, and site occupancy, benefits of two-dimensional LC-MS for the characterization of ADCs, and finally, a quick presentation of the bioanalysis of antibody drug conjugates. The methodologies we developed were applied to 2 gold standards corresponding to 2 ADCs currently on the market: Adcetris, or brentuximab vedotin, an ADC conjugated on cysteine residues, and Kadcyla, or trastuzumab emtansine, conjugated on lysines. So let's move now to the first part of the webinar with the characterization of ADCs at top level for the determination of the drug-to-antibody ratio. The drug-to-antibody ratio, or DAR, is a critical quality attribute of ADCs. Low DAR species have a low efficacy, while high DAR species can lead to safety issues, among which problems of immunogenicity. DAR determination of lysine-linked ADCs is routinely performed by UV spectrophotometry. However, this method is not compatible with non-UV absorbing drugs, and the DAR value can be overestimated due to the presence of free drug. Moreover, it gives the mean DAR value, but not the distribution of the number of drugs, also called drug load distribution. An homogeneous population of DAR4 won't have the same PK behavior as a mixture of species from DAR0 to DAR8 with a mean DAR value of 4. DAR determination of cysteine-linked ADCs is routinely performed by hydrophobic interaction chromatography, or HIC, with UV detection. This methodology is very efficient, but a specific method may be required for each ADC, and the separation can be tricky for highly hydrophobic drugs, for example, PBDs. As an alternative to these techniques, mass spectrometry can be used as a universal tool for DAR determination for both routine GMP work and as an orthogonal method to other techniques. The determination of DAR on intact ADCs conjugated on lysine residues is done after deglycosylation with rapid PNGase F under non-reducing conditions, which takes approximately 15 minutes. Deglycosylation is carried out to simplify the MS spectrum and to facilitate data processing. The online desalting before MS detection is done on a PRRPS column. 1 microgram is injected to perform this analysis, and the process is done automatically within the processing software. The average DAR value is calculated easily according to the formula you can see on the slide, assuming that all the species have the same ionization yield. So here is an example of Kadcyla. On the left, we have the raw electrospray mass spectrum with the envelope of multi-charged species, and on the right, the deconvoluted spectrum with species ranging from DAR0 to DAR8. If the amino acid sequence and the elemental formula of the drug are entered into the software, each peak can be identified, taking into account post-translational modifications, such as lysine clipping if applicable, and the DAR value is calculated. A value of 3.8 is obtained, which is in agreement with the value obtained by UV spectroscopy, which is 3.9. The methodology is different for the methodology is different for cysteine-linked ADCs due to the partial reduction of interchain disulfide bridges. So non-denaturing conditions, also called native conditions, have to be used to avoid the disruption of non-covalent interaction. As before, deglycosylation is performed, and the desalting is now done in native conditions by size exclusion chromatography using ammonium acetate at neutral pH as the mobile phase. The ion source conditions of the mass spectrometer also have to be optimized to maintain the non-covalent interaction in the gas phase, and we have to inject 20 micrograms as native MS is less sensitive. The example of Etcitris is shown on this slide. So the raw electrospray spectrum is less complex as the protein is less charged, and charged states are then shifted to higher M/D values. The deconvoluted spectrum shows species ranges from DAR0 to DAR8, and the average DAR value calculated is 3.9 in good agreement with the value that we can obtain by hydrophobic interaction chromatography. If we now want to go further into detailed characterization, we can also use a middle-level approach. Intact mass spectra are very useful and easy to obtain, as I shown earlier, but they only give a global view of the ADC. Reverse phase LC-MS analysis at the subunit or fragment level also has some advantages. A better mass accuracy is obtained due to the lower molecular mass of the species that are analyzed. For the same reason, a better chromatographic resolution of variants can be obtained. and it is also possible to get information on the localization of the conjugated drugs. There are two options for these studies. The first one consists in ADC denaturation and reduction, for example with DTT, and the second option involves a digestion by IDS enzyme and further reduction with DTT. IDS is an enzyme that cleaves around the hinge region of the ADC to yield Fab and Fc fragments. After reduction, all the fragments obtained have a molecular mass close to 25 kilodaltons, which is easy to analyze by LC-MS. The sample preparation is quick, about 1 hour and a half, and is compatible with most formulation buffers, so you do not have to remove your buffer before performing the analysis. However, the choice of the LC column is critical to get correct results in LC-MS. We have compared different columns on an easy example, a monoclonal antibody digested by IDS without reduction, and you can see that the profiles are really different from one column to another, and the chromatograms obtained with the last column give excellent results and allow the detection of minor variants that are not visible with other columns. This chromatogram gives the example of Kadcyla, so a lysine-linked ADC, studied by LC-MS after reduction. Main peaks can be identified based on MS data, but the heterogeneity of the conjugates is so high that the more hydrophobic species cannot be resolved chromatographically. The same kind of results would be obtained after IDS digestion. The subunit analysis of lysine-conjugated ADCs is therefore not really useful. By contrast, chromatograms obtained for cysteine-linked ADCs are very clean. Here is the chromatogram obtained for red citrus after IDS digestion and reduction. Each peak is identified thanks to a nice mass spectrum, and the mean DAR value can be calculated based on UV detection. This type of analysis can be used for characterization, but also for routine analysis or comparability studies. The third part of this webinar will now deal with the bottom-level approach, more widely known as the peptide mapping approach. As for other therapeutic proteins, peptide mapping is a method of choice for the confirmation of primary sequence, the characterization of PTMs, including glycosylation, and targeted purity profiling studies for the monitoring of degradation such as oxidation or deamidation. For ADCs, peptide maps can provide additional information on the precise localization of conjugation sites and the determination of site occupancy based on MS data. UPLC combined with high-resolution MS and dedicated software allow the use of this methodology even in a routine work. The classical method we developed involves the use of a double digestion with trypsin and glucine overnight. However, the peptide mapping of lysine-linked ADCs requires specific conditions to determine conjugation site occupancy, as trypsin cannot cleave after conjugated lysine residues. Therefore, 2 digestions are required to get both good sequence coverage and site occupancy information. Therefore, we decided to use trypsin plus glucine and aspen plus glucine as a mixture of enzymes. Optimization of the method was necessary as regards the choice of the enzyme, the mobile phase, the gradients, and the MS conditions in order to be able to detect all peptides, even the most hydrophobic ones. A UPLC separation over a 2 hours gradient was used, with MS detection on a QTOF mass spectrometer. Data were processed automatically within the processing software. The chromatogram you can see here corresponds to the peptide mapping of citrus obtained with a trypsin plus glucine digestion. A nice resolution is obtained between the different peptides. And as you can see on the coverage maps, the sequence of coverage is really close to 100% with only 3 amino acids missing. I will now focus on this peptide that you can see on the sequence, containing 3 cysteines, which will be very hydrophobic if all the cysteines are conjugated to a drug. We can see that the method is able to detect this peptide, which is marked with a star on the chromatogram. To confirm the identification, we have studied the high energy spectrum in MS to the E mode, and the ion at M/Z 718 and the successive losses of 3 drugs confirm the identity of this peptide, bearing 3 monomethyl auristatin E residues. Kadcyla was then used as a model for lysine conjugated ADCs. In that case, we have two digestions, one with trypsin plus gluci, and the second one with aspen plus gluci. And as you can see, both maps can give very satisfactory resolutions, and the second coverages we obtain are presented here. The coverage is 99% for trypsin plus gluci and 95% for aspen plus gluci. This shows the complementarity of the two digestions. Trypsin plus gluci can be used to get an excellent sequence coverage, for example, to confirm the primary sequence, and aspen plus gluci can be used to determine the site occupancy of the different lysine residues. So in this slide, you can see once again the peptide map of Kadcyla obtained with aspen plus gluci. And as the conjugation of lysines yields a very heterogeneous mixture of species, it would be interesting to be able to detect specifically the conjugated peptides. This would be a nice tool, for example, for comparability studies. So to do that, we studied the fragmentation of the conjugated peptides, and as we see here, we detect two specific fragment ions at M/Z 485 and 547. Based on DM1 structure that you can see here, we propose the following molecular structure for these ions. And if we extract those ions specifically from the high energy trace, we can easily detect all the conjugated peptides. This methodology can be used for comparability studies, for example, to check that the conjugation profile is similar from one batch to another. In some cases, advanced characterization with precise identification of the conjugation sites can also be necessary. Some conjugated peptides may contain two or more lysine residues, and MS alone will not give any information on which residue is actually conjugated to the drug. As shown on this MSMS spectrum, using regular MSMS or CID for collision-induced dissociation, the fragments that are detected correspond to the drug loss only, and there is no information about the amino acid sequence. A solution is to use electron transfer dissociation, or ETD, which cleaves the polypeptide backbone without fragmenting the drug. The ETD spectrum shown here was obtained on an ion trap mass spectrometer. Information on peptide sequence is obtained, and the drug can be localized unambiguously on the first lysine of the peptide. The fourth part of this webinar will deal with the characterization of antibody-drug conjugates by 2D LC-MS. We focused here on the use of hydrophobic interaction chromatography in the first dimension, which is a method of choice for the characterization of antibody-drug conjugates. It is mainly used for the determination of naked antibody for lysine-linked conjugates or site-specific conjugation technologies, but also for the determination of the drug-to-antibody ratio and drug load distribution for cysteine-linked conjugates. Due to high salt content in mobile phase, it is impossible to effinate the chromatography to MS for peak identification. A solution is to use fraction collection and desalting, but it is really time-consuming and not easily amenable to low-concentration species due to dilution effects. Therefore, 2D LC-MS is a solution for a quick identification of the peaks that you can observe on Higgs chromatograms. A few words on the principles of two-dimensional liquid chromatography. So in the first step, the system is used as a 1D LC system with only optical detection. In the second step, when the peak of interest elutes, the valves are switched and the eluate of the first column is sent to a trap column. Here we use the C4 column as the trap. This methodology is called herd cutting, and this step is usually 6 to 7 seconds long. An isocratic solvent manager is used to send water to the trap column to increase the desalting efficiency. In the third step, the trapped sample is sent to the second dimension, and the desalting and the separation is done on a reversed phase column. This is a fully automated process which provides a very robust and efficient desalting. This is applicable to hydrophobic interaction chromatography, but also to any other separation, such as ion exchange or size exclusion chromatography, which contain a huge amount of salt, which is not amenable to mass spectrometry detection. So here is an example of a Higgs separation we developed for ADCs. As you see, a double peak is observed for what we expect to be DAR4 species, as you can see with the stars on the chromatogram. We have analyzed those two peaks by 2D LCMS, and we were able to confirm first that we had two DAR4 isomers, and then we were able to identify those isomers. For the first one, on the second dimension, we observed two species as the non-covalent interactions are broken in the reverse phase condition. For the second peak, only one species is detected, and therefore the two isomers can easily be identified as isomer 4A and 4B, as you can see on the screen. By analyzing all the peaks, we were able to identify all the species, either the minor species such as DAR1 and probably DAR3, even if the signal in mass spectrometry was quite low. Once all the species are identified, the relative intensity in UV can be used to calculate the average DAR value. In that case, we obtained a DAR value of 3.7. So let's finish now this webinar with a few words about the bioanalysis of antibody-drug conjugates and the role of mass spectrometry for these studies. Bioanalysis of antibody-drug conjugates is much more complex than the bioanalysis of traditional protein drugs. Due to the complexity of the molecule, additional tests are required to evaluate the stability of the conjugate in the circulation. As you can see here in this table, you have a typical analytical package for pharmacokinetic studies. The conjugated antibody is usually quantified by ligand binding assays, typically ELISA. Total antibody can be quantified either with ELISA or LC-MSMS, depending on the methods that are available and the stage of development you are in. The drug conjugated to antibodies can also be quantified after cleavage from the ADC by affinity LC-MSMS or ELISA. The free drug, which is a critical analyte to be quantified due to its high toxicity if it is released from the ADC, is quantified by LC-MSMS, as it is typically done for small molecules. And finally, anti-therapeutic antibodies that have to be quantified to evaluate the immunogenicity of the antibody-drug conjugate are analyzed using a ligand-binding assay. Finally, an additional test can be used to evaluate the stability of the ADC, and it is the determination of the drug-to-antibody ratio in plasma, which is done also by LC-MSMS after immunoaffinity. So I will now describe two techniques, which are the determination of the total antibody by LC-MSMS and the determination of the drug-to-antibody ratio also by LC-MSMS. So in this slide, you have the strategy that we use to quantify the total antibody in plasma during preclinical studies by LC-MSMS. So first, we had a fully labeled antibody to the sample as an internal standard. This internal standard will have a role in order to take into account any loss that could occur during immunopurification and any variability during the digestion of the antibody and its analysis by LC-MSMS. Then we isolate the antibodies of interest, so the ADC plus the internal standard by immunoaffinity on magnetic beads, using a coating of either protein A or anti-human antibody if we need increased specificity. Then we reduce and alkylate the sample before digestion with trypsin. And finally, the antibody is quantified by LC-MSMS using 2 to 4 signature peptides. The sensitivity of the method, as you can see here, the lower concentration we analyze is 0.1 microgram per ml, but it can be lower if necessary. And this sensitivity is perfectly compatible with the concentrations that are typically found in preclinical studies. So as I said earlier, the determination of the drug-to-antibody ratio in plasma can also be useful to evaluate the stability of the conjugate during preclinical studies. To do so, we isolate the antibody-drug conjugate from plasma using magnetic beads, coated either with protein A or an anti-human antibody. and then we elute the ADC and we inject it into the mass spectrometer using a short online desalting step. All the protocol can be performed in a single 96 well plate, which can be very useful if you have a huge amount of samples to analyze simultaneously. So the result presented here with Kapsila was obtained without deglycosylation, but an optional deglycosylation step can be added to simplify the spectra and increase the sensitivity if necessary. As a conclusion to this webinar, I hope I have convinced you that mass spectrometry is a valuable tool for the characterization and bioanalysis of antibody-drug conjugates. Advances in hardware and software now allow the use of MS for routine analysis in a GMP or a GLP environment for preclinical studies. Characterization can be performed at different levels for both cysteine and lysine-linked ADCs, and of course for site-specific conjugates. At the intact level for the determination of the drug-to-antibody ratio, we can obtain both the mean value and the drug load distribution. At the subunit level for mean DAR value and localization of the drugs on the different fragments. And at the peptide level for the localization of the conjugation sites and the determination of site occupancy. 2D LC-MS is also a great tool for the identification of species detected in hydrophobic interaction chromatography and UV detection, but also for ion exchange or site exclusion chromatography. And finally, as you can see, MS is also a key element for the bioanalysis of antibody-drug conjugates. We have optimized workflows and methods that are robust and reproducible, sensitive, fast, versatile and customizable to any need you may have, depending on the chemistry of conjugation or the drug that is linked to your antibody. They can also be used routinely in a regulated environment thanks to validated software that are now commercially available. And finally, the process and reporting are also automated and user-friendly to save time. All these methods can be used for quality control, comparability studies, process optimization, or stability testing. And they can also be used as orthogonal methods to more classical techniques in order to confirm the results and, if necessary, validate those classical techniques. Quality assistance can propose a testing strategy that best suits your needs and also optimize and validate the analytical methods that are dedicated to your product according to ICH guidelines. I want to end by thanking all the people that were involved in this work, and more specifically Eric, Anissé, and Fabrice, who performed all the analytical work that I presented today, and all the staff at Quality Assistance. And I also thank the Région Wallonne for funding. I thank you for your attention. I will be pleased to answer your questions.
Thank you very much, Dr. Delobel. So at this point, I would like to begin the question and answer session. Just as a reminder to our viewers, you can ask questions via the Questions tab located at the bottom of your screen, and Dr. Delobel will happily answer your questions. So please send them through to us. So our first question to Dr. Delobel is it required by the guidelines to determine the drug load distribution to release testing of ADCs?
So there is no guideline currently available specifically for antibody-drug conjugates, so people have to rely on the guidelines for small molecules and biologics. But the FDA gives some presentations in different conferences to tell exactly which techniques should be used. From what I heard recently, the drug load distribution is not something that has to be included in the analytical package. However, it's very, very useful for supporting information, and it's highly recommended by the FDA currently to perform drug load distribution testing, even if it is not included in the release package and even if you don't have specifications for this test.
Great, thank you. So the next question is, to determine the drug-to-antibody ratio, you need to assume that all species ionize the same way. Is this really true?
You need to, if you want a real value, an accurate value, the species have to ionize the same way, or you have to correct by the ionization efficiency. unfortunately, it's almost impossible to verify the ionization yield, because to do that, you would need isolated DAR0, DAR1, DAR2 species, which is very difficult to achieve. So the best way to prove that the different species ionize the same way is to use an orthogonal technique such as UV spectrophotometry for lysine link conjugates, or hydrophobic interaction chromatography for cysteine link conjugates. And if you get similar results using both techniques, you have demonstrated that the ionization yield is similar. So that's the best way to do that. But people that can have access to isolated species for DAR0 to DAR8 can demonstrate that the ionization efficiency is similar, and that would be the best way to perform that.
Thanks, Dr. Delobel. Just as a reminder to our audience, if you have any questions for Dr. Delobel, please send them through via the questions tab at the bottom of your screen, and we will ask Dr. Delobel for you. So our next question is, don't you think that for routine analysis, you can skip the use of MS and use only hydrophobic interaction chromatography?
Yeah, it's clear that MS, especially 2D LC-MS, as I showed in this webinar, is used mainly for investigations. So once you have developed your hydrophobic interaction chromatographic method, you perform 2D LC-MS to identify all your peaks, and then once everything is identified, you can rely on UV detection and peak intensity in order to perform the DAR calculation. However, for some species, so if you have very hydrophobic payloads, you may not be able to use hydrophobic interaction chromatography. You won't have sufficient resolution between the different isomers. And in that case, you won't need 2D LC-MS, but you'll have to switch to intact mass measurements in order to perform the DAR determination and drug load distribution determination.
Thank you very much. Our next question. Can you use deconvolution algorithms such as MaxEnt in a regulated environment?
That's a question that we had quite often because the algorithm that is used to deconvolute the raw electrospray mass spectrum is quite complex. It's impossible to verify the results by hand. And it's true that if you change just a little bit the parameters that you use for the deconvolution, the molecular mass you measure will be quite different. So, but as far as you use the same algorithm and the same deconvolution parameters, in our experience, the mass accuracy you obtain and the precision on your measurement is very, very high. Second point is that in what I showed here, the accuracy of the molecular mass is not critical. What you need is to be able to differentiate between DAR0, DAR1, DAR2, etc. And in that case, as far as you have the resolution between your different species and you have the convolution that gives you the correct amount, relative amount of the different species, even if your mass accuracy is not perfect, you will be able to determine the DAR value with a very high accuracy. So MaxEnt can definitely be used in a regulated environment, but you have to be aware of the deconvolution parameters you use.
Thanks very much, Dr. Delobel. So that seems like all the questions that we have today. I would like to take this opportunity to thank Dr. Delobel for sharing his knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com/webinars. Thank you very much, everyone. Thank you, Dr. Delobel.
Thank you.